Hello, everyone. In this chapter, we are going to address gene expression and epigenetics. We are going to dive into how gene expression is controlled over time in tissues, cells, and organs. So, as we address this chapter, I want you to focus on a number of questions. The first question is going to be, how does gene expression vary over time and based on the tissue type? How is gene expression controlled? You know, for instance, that we talked about a muscle cell being different from a nerve cell, not only in terms of structure, but also in terms of function, right? While having the exact same genetic information, 100%. And we did say that differential gene expression is what causes specialization of cells, right? So the pattern of proteins that are going to be found in different cells is going to be different, right? So these are the kind of aspects we are going to dive into in this chapter. Another question is going to be, how do exons and introns allow the cell to maximize the number of protein produced? In other words, we realize as we look at the number of genes in the genome and the number of proteins that are produced, there is no exact match. We don't have as many genes as proteins and vice versa. And what can account for that difference, right? Now, let me tell you upfront that in our body, we have much more proteins than we have genes. But we know in the central dogma that we go from DNA to RNA to proteins, right? So technically, one gene produces one messenger RNA, which produces one protein, but we have more proteins than number of genes, right? So we are going to take a look at that. And then we are going to discuss high viral DNA, non-coding RNA, and repeated sequences of DNA account for a large proportion of our human genome. So let's take a look at the first step, which is how gene expression changes over time and based on tissues. All right, so first and foremost, know that the genome functions like an orchestra, right? Where you have different instruments. And when I say different instruments, I'm referring to different genes, right? Different organs, different cells, different tissues. So, in spite of the fact that different cells, different tissues, different organs are going to look different and be different, nevertheless, it's the same genome, meaning the same orchestra. So we notice that not all genes are expressed continually all the time in different cells, in different organs in the body. However, it is important to mention that some genes are always expressed in all cells. For instance, all cells need ATP to function. Without ATP, a cell is not going to function because ATP is the currency of energy in the cell that allows cellular activity to take place, which means that all cells will have to produce ATP. That's why all cells need mitochondria in our system, because we are aerobic organisms, right? Using aerobic metabolism, right? So we have a set of genes that are involved in orchestrating the basic tasks that should happen in a cell, like to nourish the cell, to survive, to reproduce, right? Now, these sets of genes are called housekeeping genes. These housekeeping genes are the ones that are going to do the basic functions. Now, when it comes to specializing for a given cell, a muscle cell that is different from a nerve cell, obviously different in shape and different in function, now something has to make them different. So some genes are going to be turned on in a nerve cells that are not turned on in a muscle cell, and vice versa, right? So the instruments, like in an orchestra, the instruments are not playing at the same intensity at the same time. Sometimes there is a couple of instruments that will have to be louder than other instruments. Other times, we want all the instruments to participate with an equal intensity, right? So this is very important to keep in mind. That analogy is very important to keep in mind, right? So differential gene expression is going to occur because some genes are allowed to be expressed in some cells while not allowed to be expressed in other cells, right? Causing different sets of proteins to exist in one cell and not exist in another cell. For this can happen at a particular time, but this can also happen at a particular space, right? So space and time play a very important role. And when I say space and time, I'm thinking, for instance, during the development of an embryo, right? You have fertilization, then you are an embryonic stage. From an embryonic stage, you switch to a fetal stage of development, and from a fetal stage of development, then an individual is born, and that individual is going to go through a number of experiences. They will start eating, they will start encountering some infections, they will start experiencing some environmental conditions such as radiation from the sun, and so on and so forth, right? All of that is going to affect how genes are being expressed, right? Now, when we look at the expression of the genome in a given cell, a given organ, a given tissue, right? All the transcripts in a given condition and time is called a transcriptome, right? So these are all the transcripts, right? If we focus on a cell, it's going to be all the transcripts in a cell. But we can be interested in knowing what are all the transcripts in a given tissue at a given time, right? That would also be a transcriptome. All right. So gene expression changes over time and in different cell types. I already mentioned that, right? This change can occur at a very molecular level based on the tissue, based on an organ, or at a gland level, right? Now, we are going to notice that the changes that occur in gene expression are mainly based not on the sequence of DNA in the genome, but on little things that attach to DNA or to proteins that are associated to DNA, which altogether are considered epigenetic changes. Okay, these are the change to the chemical groups that associate with DNA and that can be transmitted to daughter cells after cell division. But not always, right? Not always. So when we look, for instance, at the the placenta, and we look into the genes that are expressed in the placenta, we notice that, well, the transcriptome is going to vary based on space and time. Now, the discipline that studies the transcriptomes in a tissue and organ, it's basically called tissue transcriptomic or an organ transcriptomics, right? So this allows us to know the physiology of a particular organ at a particular time, right? In this particular instance, we are going to focus on the placenta. So in the placenta, when we look at gene expressed, we're going to notice that the genes that are expressed in the placenta fall into 5 groups. Out of these 5 groups, only one group represents mom's DNA, mom's RNA, actually, mom's messenger RNA. And this messenger RNA that comes from mom actually are genes that, when expressed, are going to cause mom to tolerate pregnancy. So they're going to dampen mom's immune response so that mom's body doesn't reject the growing new life in them, right? So this sets of genes are from mom. Otherwise, the 4 other groups of genes are genes that are going to allow the implantation of the fetus, the nourishment of the fetus, and also the release of hormones that allow mom to maintain pregnancy. So basically, the newly developing organism is going to run a number of genes that are going to express so that mom can tolerate pregnancy. So actually, when we think about that, there are really 2 levels of actions that allow mom to maintain pregnancy. One is that mom herself is expressing genes that purposefully causes mom to be somewhat immunocompromised, right? So the immune system decreases so that the growing fetus is not rejected, but also the fetus itself is expressing some genes that causes mom to tolerate all of that. And this is beyond nourishing the fetus and causing the development of the fetus. All right. So let's take a look at a single cell, and let's take a look at the transcriptome in a single cell. That is another level of discipline in biology that is called single cell transcriptomics. In a single cell transcriptomics, we are going to focus on one single cell. Previously, we focused on a tissue and an organ, right? Now, we are focusing on one single cell. What we notice is that when it comes, for instance, to the production of hemoglobin, we notice that, well, that hemoglobin is going to change over time during the development of the fetus and during the life of an individual. Now, an adult is going to have a hemoglobin molecule that is made up of 4 chains of globins, right? Now, each chain of globin is going to have a hem group that has iron, right? So the hem group has iron, right? Now, as this hem group with an iron atom combine with the globin, this forms hemoglobin. And you need 4 hemoglobin molecules, 4 subunits, to actually be functional. Now, we have a beta chain of hemoglobin, another beta chain of hemoglobin. So we have 2 beta chains of hemoglobins. A beta chain of hemoglobin is going to be made up of 146 amino acids, and the genes responsible for the production of the beta chain of hemoglobin are carried by chromosome 16. Okay? The second chain of hemoglobin is going to be the alpha chain. Now, that alpha chain the gene for alpha chain is going to be carried by chromosome 11, right? A functional hemoglobin in an adult is going to have 2 alpha chains. Each of the alpha chain is made up of 141 amino acids. So a functional hemoglobin molecule to carry oxygen will need to be formed of 4 units that are folded in 3D to form a quaternary conformation of the protein. Out of that quaternary functional protein, you are going to have 2 alpha globins and 2 beta globins forming hemoglobin. All right, now diving a little bit now into the change of the hemoglobin composition over time, here is what we see. We see that there are 3 forms of hemoglobin that occur from the point of inception to adulthood. We have embryonic hemoglobin, we have fetal hemoglobin, and we do have adult hemoglobin. Now, embryonic hemoglobin is the hemoglobin that is prevalent during the embryonic stage of development. So here, the hemoglobin is going to be made up of 4 chains in terms of embryonic hemoglobin. We have 2 chains that are epsilon chains and 2 chains that are zeta chains. Now, we have epsilon chain and zeta chains. What we notice is that at the embryonic stage, while we have epsilon and zeta, the amount of epsilon and zeta is going to decrease over time. And when we are at about 6 weeks after fertilization, beyond 6 weeks after fertilization, we don't have any more embryonic hemoglobin, right? We have a loss of zeta and epsilon hemoglobin chains. While this is happening, notice that the gamma chains start being produced before the embryonic hemoglobin is gone, right? Gamma start being produced, right? And along with gamma that start being produced, you have alpha hemoglobin that start being produced. Eventually, there is a point in time when from beyond 7 weeks after fertilization to the day of birth, where fetal hemoglobin is going to be the prevalent hemoglobin. And that fetal hemoglobin is made up of 2 gamma chains and 2 alpha chains. And you see that the gamma chains and alpha chains are going to persist all during pregnancy until birth. Now, beta hemoglobin does not start being produced until close to birth and after birth. Now, close to birth, we see that the amount of beta globulins is starting to increase rather exponentially, while the amount of alpha is drastically decreasing, depleting, right? While alpha hemoglobin is staying constant. Now, eventually, and notice that after birth, right, when we are about 12 weeks after birth, almost all the gamma hemoglobin is gone, right? Which means that there is a progressive switch from fetal hemoglobin to adult hemoglobin. In fact, by the age of 4, all hemoglobin that is found in a person is basically made up of adult hemoglobin. And that adult hemoglobin, it's 2 beta chains and 2 alpha chains. About 99% of the hemoglobin is adult hemoglobin by age 4. Okay? So we see that the composition of hemoglobin is going to switch based on the level of oxygen. Now, why do I say this? I say this because in an early stage of development, at an embryonic stage, the zygote And further, the the morula and the blastula, are not as exposed to oxygen as when a baby is born, right? And start breathing air, right? Oxygen in an embryo depends very much of the amount of oxygen that mom is providing, right? At a fetal stage, the baby is starting to, the fetus actually is starting to have organs and blood vessels, and these blood vessels are going to produce blood, right? And that blood is going to carry oxygen. So the more the fetus develops into becoming a perfect human, the more oxygen is going to be needed, right? Which is actually going to switch the composition or the makeup of the globin chains. When the individual is actually born and starts breathing, this is now where the individual has to figure out on their own how to make good use of all the oxygen that they are breathing. And so the makeup chains has to also switch, right? So this speaks to the different gene expression for the production of globulins in during the development of an embryo, the fetus, and the growth of an individual throughout their lives. All right. So at a cellular level, looking at a transcriptome, it's relatively simple, I want to say. However, when it comes to looking at organs, the story becomes more complicated. It becomes more complicated because an organ is solid, right? An organ is solid, and an organ has a specific shape. And that specific shape is controlled by many, many environmental conditions, right? For instance, a liver is not shaped like a stomach. That is not shaped like a heart. That is not shaped like a kidney, right? So, at an early stage of development, when differentiation starts happening and when we start seeing the future arms, the future legs, the future kidney, the future lung, you have a level of differentiation that starts there. But then you need, or the body needs to maintain that differentiation so that the heart never becomes the kidneys, right? That the kidneys never turn into digestive organ, right? For instance. So all of that means the expression has to be tightly orchestrated to maintain a certain direction of differentiation, but also to maintain a number of cells to stay blank cells, right? So while allowing some cells to undergo differentiation, other cells have to absolutely remain blank cells, right? Which means that, well, we have to tightly control what is being expressed when, so that there is no confusion. Now, you understand the level of complexity we are going into right now, right? So the genes are going to be turned on and off during the development. Some genes are expressed at an early stage of development that should not be expressed at a later stage of development, right? And vice versa, right? Because we don't want to see aberrations, all right? Stem cells have to self-renew and keep that integrity, right? And eventually, some have to specialize, right? Okay? Now, one example we are actually going to take a look at is the organization of the pancreas. The organization of the pancreas is relatively simple, right? The pancreas is an organ that has two functions. It's an exocrine gland and it is an endocrine gland. Exocrine because it's going to produce all the pancreatic juice, and that pancreatic juice is going to be dumped into the small intestine, specifically the duodenum area, right, where much of the digestion is going to happen. But you have other part of the pancreas that is specialized in producing hormones. One of the most well-known hormone is insulin, right? The second one is glucagon. They are both involved in controlling the amount of sugar in the blood, right? These hormones are not dumped in the small intestine like the pancreatic juice is, but these hormones go in the blood, they are released in the blood, and from the blood they're going to travel, right? That makes this endocrine, right? An endocrine function. All right. So, when looking at the cells that at the end of the day make up the pancreas, they start as stem cells. These stem cells will have to make sure that we always have stem cells that are blank all the time, right? So that from these blank cells, differentiated cells can be produced. So now, out of the stem cells, you are going to have some cells that are going to start their differentiation, but as they start their differentiation, they have two possibilities later on. But before they develop these two possibilities, they first have intrinsically the features that allows them to become one or the other. So these are the exocrine endocrine progenitor cells. This exocrine endocrine progenitor cells are going to further, based on the kind of signal they receive, to turn into exocrine progenitor cell in one hand and endocrine progenitor cells on the other hand, right? The exocrine progenitor cells are going to mature into exocrine cells that are actually going to produce the pancreatic juice. While the endocrine progenitor cells are going to differentiate, and again, this is based on the kind of signal they receive. They're going to differentiate into beta cells. These beta cells produce insulin, okay? And the beta cells represent about 70% of the endocrine cells in the pancreas. Then we are going to have about 15% of the cells that are going to turn into alpha cells. Okay? These alpha cells are going to produce glucagon, while insulin allows the blood sugar to decrease. So insulin is going to foster internalization of glucose to go in the liver, in the muscle, to form glycogen, right? While glucagon is going to do the opposite job. It's going to target liver cells, muscle cells to break down glucagon and release single molecules of glucose. And these single molecules of glucose are the ones that are going to travel and go in the blood and be distributed through the body so that we don't fall into hypoglycemia, right? Now, some other endocrine progenitor cells are going to turn into delta cells. These delta cells are going to release a hormone that is somatostatin, okay? And these cells represent about 10% of the endocrine cells in the pancreas. Somatostatin is the hormone that controls appetite, right? And finally, we're going to have 5% of the endocrine cells that are going to represent F cells. These F cells are going to be responsible for the production of pancreatic polypeptides, okay? So these are basically the differential gene expression that is going to happen in the development of the pancreas. Now, transcriptomics allows us to identify all the messenger RNA that are in particular cells, particular tissues at a particular time. So really based on the conditions, right? So that's one thing. The other thing is going to be proteomics. Proteomics does the exact same thing, except that rather than focusing on the messenger RNA, with proteomics, we are going to focus on the actual proteins that are produced. Which proteins are present in a muscle cell at a given stage of life? We can ask the same question for which proteins are all present in a nerve cell at a particular stage of your life, right? In a particular condition of your health, right? So transcriptomics and proteomics are very useful to understand the biology of an organism as it develops, right? A different stage of development. So because of transcriptomics and proteomics, we can actually understand why some diseases develop, because when looking at a particular disease, we can notice that some proteins are more abundantly present in the body than in a healthy individual, so that we can actually ask, okay, well, let's just restrict proteomics into when someone is sick, right? So we are going to actually look at genes that are expressed, proteins that are produced in a sick person compared to a healthy person. This actually is called diseasome, okay? diseasome. Say, for instance, someone experienced a heart attack. Okay? So when someone experiences a heart attack, what is the proteome at that point in time, right? When someone is expressing obesity, right? What are all the proteins that are characteristic of obesity, right? So we can now, by studying diseasome, connect a particular disease or a group of disease with some genes that are expressed, right, in that condition, right? Many studies were done with obesity, hypertension, and diabetes. Now, others have been rather surprising. For instance, in the case of heart attack and in the case of the Duchenne muscular dystrophy, which is a wasting disease, right? So now researchers are able to test heart drugs based on the diseasome in people with heart attack. Also, they are designing drugs based on the diseasome in Duchenne muscular dystrophy, right? So here are some examples of diseasomes. Now, you see that it's kind of like a network, okay? It's a network. Say, let's focus on heart disease. So we notice that heart disease can be associated with genes that are expressed in a case of deafness, in a case of cataract, in a case of muscular dystrophy. So we see that there are some genes that are expressed that are commonly found in these different conditions, which means that sometimes when we design a treatment for one condition, that treatment may or may not work for an associated condition. In other words, we can design a drug to treat heart disease, and that drug, we can find out that that drug is actually working also for muscular dystrophy. And we can be like, this is really two different conditions, right? Yet the drug is going to work in the extremely different conditions, right? So this is basically a way to see how we can actually make a best use of the drugs we have, right? Alzheimer disease, there are some genes that are found in Alzheimer disease that has been associated with heart attack, with diabetes, with schizophrenia, with hypertension. And so you can do the same thing with many other diseases, right? To actually come up with a diseasome.